Hello everyone, welcome to Unit 2. In this unit we're going to cover chapters 7, 9, 10, 11, and 12. It is divided into 3 parts, so very similar to Unit 1. Please take your time, go through the slides, listen to the audio, and if you have any questions, comments, and concern, please keep in mind that I'm always available through email, use your discussion session, or if you'd like we can set up a Blackboard Collaborate. All right, so without further ado, let's start taking a look at Chapter 7. Now, hopefully you guys remember the law of independent assortment, right? We talked about this in our previous chapters in Unit 1, and based on Mendel's work, the law of independent assortment basically said that when you're looking at 2 genes, when it's time to go from diploid to haploid in forming your gametes, the genes do not automatically have to travel together. And this then led us to our formation of our dihybrid Punnett square, where we were doing our foiling technique to come up with all the possible combinations that the genes can move from diploid to haploid. Now, it turns out that not all genes will adhere to Mendel's law of independent assortment. There are genes out there that are physically linked together because they're located on the same chromosome, which kind of makes sense because if you think about it, humans, we have 23 pairs of chromosomes, but based on research, we know that we have anywhere between 22,000 to 23,000 genes. That is a massive amount of genes to kind of compile within a short amount or a small amount of chromosomes. So we see that each chromosome has the ability to carry hundreds, if not thousands of genes, which would go without saying that some of these genes are so closely linked together that they have no other option but to move together when you go from diploid to haploid. And if that is going to occur, then that will mean that you no longer follow the rule of Mendel's law of independent assortment. So let's, for instance, take a look at this example right here. So we are looking at 2 specific traits at this flower. We're going to take a look at the color, and we're going to take a look at the pollen. So go ahead and start taking a look at your homozygous mix that's on the left-hand side in a parental generation, and you can see that you're looking at a homozygous dominant mix for both traits that will include the purple flowers and the long pollen, okay? So we have our homozygous dominant traits for 2 of the characteristics on our left-hand side. And on the right-hand side, we have our homozygous recessive traits showing that we have our homozygous lowercase p's for the red flower coloration and the lowercase l's for the round pollen. Now, based on what we know is that if you go ahead and you create your dihybrid Punnett square and you go in with homozygous mixes for both genes, you know that no matter how you twist or turn, the gametes for parent 1 will be capital P, lowercase, capital L, excuse me. Those are your only options of how they can move together. And if we take a look at parent number 2, we know that all the possible combinations for the gametes will include lowercase p with lowercase l. And if we plug that into the Punnett square, then what we see happening is that all of our offspring will then be heterozygous when it comes to both of the traits. So you can see we have our heterozygous for the purple flowers, and we have our heterozygous for the long pollen. Now, remind yourself, right? So you have your heterozygous mix. Let's go ahead and do a self-fertilization. So we are going to go ahead and take the 2 heterozygous parental strands, and we're going to go ahead and create our F2 generation. Now, what is the stereotypical ratio that you would expect if you were taking a look at heterozygous organisms, right, for the 2 traits? We talked about this previously. And if you want to remind yourself, take a look at the end of the table right here. We can see that the expected ratio, if we're doing 2 heterozygous parental strands, is then going to be for the 2 traits is that we expect a 9 to 3 to 3 to 1 ratio, okay? Now, let's go ahead and do this experiment in real life according to this picture. So they went ahead and they did the self-fertilization with the heterozygous mix. And if you take a look at the table, you're going to notice that they have the observed number. So this is what they actually saw compared to the expected number. And the expected number is all based on Mendel's law of independent assortment that tells us that we should have a ratio of 9 to 3 to 3 to 1. So go ahead and take a few seconds and compare the numbers that were observed versus the expected number based on the ratio from Mendel's prediction. Alrighty. So hopefully as you're looking at it, one thing that's going to peek out of you is that there was a much greater proportion of the two types found in the parental generation than you would technically be looking for. What do I mean with that? Well, go ahead and compare your observed number for the purple flowers with the long pollen. We were able to observe over 296, so close to 300, where we were only expecting 240. And the red pollen, the red flowers with the round pollen, we were only expecting to see 27, where they saw over 85 of them. So how do you explain these numbers being so much higher than you normally would see? And one of the possible explanations is that the purple flowers and the long pollen are not independently assorting. Instead, they're actually traveling together. The same goes with the red flower trait and the round pollen, that these two traits will tend to be linked together. Now, that means that Mendel's law of independent assortment is not applying, and therefore we are not seeing the observed number matching our expected number ratio of 9 to 3 to 3 to 1. Now, if you're seeing this happen, that the genes tend to travel together as a couple or as a group, we can then go ahead and say that these genes are linked together, or that they form a linkage. And that indicates that they tend to be found on the same chromosome, relatively close together, and when it's time for assortment to occur and the organism to go from diploid to haploid, these particular genes will then travel together as one unity. We can also see, for instance, in humans that we have different linkage groups. So for instance, our autosomes, our autosomal chromosomes, they actually form 22 different linkage groups, one for each of the pairings, where the maternal and the paternal chromosomes are linked together. So chromosome number 1 is one linkage group, 2 is the second, 3 is the third, where they encourage the movement of the maternal and the paternal chromosomes together. You also have linkage groups for when it comes to your sex chromosomes. So your X chromosome will be its individual linkage group, whereas the Y chromosome will be its individual linkage group as well. And one thing that you want to keep in mind is that when you're looking or when you're discussing a chromosome, it's really nothing more than an extremely long piece of DNA. And we basically have to create housings or loci or locations for all these different genes. So if the genes are physically linked, that usually means that they are located on the same chromosome, and they tend not to be too far away from each other. So they'll move as a unit, as a linkage, or as a linkage group. Now, the other thing you want to think about is that if you're looking at the same chromosome and you see that genes are located on it, the more distance there are between the genes, the more likely it is that these genes will go ahead and exhibit crossing over from each other. Remember, crossing over is when you mix the non-sister chromatids from homologous chromosomes and you're able to swap the maternal and parental contributions for those chromosomes. And what we see happening is that genes that are further apart on the same chromosome have a higher chance of exhibiting crossing over than the genes that are very closely linked together. Now, it turns out that if you're going to ask yourself, how often do they independently assort? This will all depend on how far apart they actually are. The greater distance allows for greater potential of crossing over between the individual genes. So what that means is that if you're looking at your chromosome, and please forgive me because my drawing ability is not the best, So let's say this is my little chromosome that I'm trying my best to draw. What I'm trying to tell you here is that if I'm looking at 3 different genes, genes A, B, and C, and what I see is that A and B are very closely located together, whereas there's a great bit of distance between C. If my organism now has to go from diploid to haploid, chances are that when it's time to separate the chromatids, A and B will travel as a linkage because they're so closely related, whereas C has a very high chance of being swapped over during the event of crossing over. So the greater the distances on the same chromosome, the more likelihood you have that crossing over will mingle the homologous genes on the chromosomes, whereas the genes that are very closely related in distance will link together. They'll move as one linkage group. And we know that this is possible because when we were discussing meiosis previously, we talked about the fact that as you go through your steps for meiosis 1 and you do your crossing over, we see that by the end of meiosis 2, when you start looking at your gametes, some of the gametes can be a parental strand versus others can be a recombinant strand. The parental strand means that they are non-recombinant cells, meaning that these are sections of the chromosomes that did not exhibit any crossing over. So they are identical to what the parental combination looked like for your individual or your mother cells or the cells that started everything off in the cell cycle. Whereas cells that exhibit crossing over, we then see that because we are mixing the non-sister chromatids on the homologous chromosome, so we're mixing the alleles from the maternal and the paternal lineage, what we then get is we get a recombinant cell, which is a genetic recombination or a hybrid of the genes that mom and dad contribute individually to the offspring. So here, we can go ahead and take a look at this from more of an illustrative example for those of us who are trying to visualize and remind ourselves about meiosis. We see right here that we have a diploid cell, and what we're looking at is a contribution from 2 parents. So we have, for instance, the maternal, and we have the paternal, and what we can see happening is that if we're going to do meiosis, our goal is to go from diploid to haploid. So one possible scenario is that we don't allow any crossing over to occur between the homologous chromosomes. We simply just break the centromere and separate the sister chromatids, and as a result, you get your 4 haploid cells that are identical copies of the original chromosome that you started with. And all you've done is you've just reduced the genetic information from diploid to haploid. So this right here would be an example of no crossing over and having no recombinant DNA being identical to the initial start, with the exception that you have reduced the genetic load. Your other option, which we know is likely in meiosis, is the fact that you will have crossing over occur. And as the crossing over occurs, then now what we get is that some of our haploid cells are going to become hybrids or are going to become recombinant, meaning that they'll be able to display both traits of the maternal and the paternal lineage. This all contributes obviously to genetic variety within the gametes, but this also allows us to explain why genes that are located on the same chromosome, if they are quite a distance away from each other, the way that you can mingle their alleles or create variety between their alleles is by utilizing crossing over. Whereas genes that are physically linked very closely together, they will not utilize crossing over. They will move as one solid unit from diploid to haploid. Now, part of the knowledge that we have from this movement of genes between crossing over and moving as a unit is all through the use of what we call genetic mapping. Genetic mapping is the concept of figuring out the exact location of genes within the chromosomes that an organism has. And a lot of this comes from completing different Punnett squares. A very popular Punnett square to complete when you're doing genetic mapping is to do what we call a test cross. A test cross is when you mix a heterozygote parent with a homozygote recessive parent. And you basically want to go ahead and predict the offspring. And as you're doing that, it allows you to see which genes are actually linked, meaning that they're moving as a unit and that obviously they're on the same chromosome. Because of genetic mapping, there is lots of information that we've been able to derive. We've been able to see how large a genome is for specific species. It has helped the mechanism of being able to clone a particular gene, to identify a location for clinical application, to understand disease models. You can also see the evolution in the sense of when we were talking about making gene families, how is the relationship different from one species to the next. And obviously, if you're interested in any type of selective breeding or control of gametes, gene mapping would be an essential tool for you to utilize. Here we, for instance, see what a partial gene mapping looks like of Drosophila. So you can see the 4 chromosomes, and the little red markings on each indicate a specific locus or location of that particular gene that we're after. And as you can see right here from the illustration, based on gene mapping, they've been able not only to tell you where the particular gene is, but they've been also able to stipulate the phenotype based on what it appears to be as a mutant and the wild type. And when you now go ahead and you start doing your test crosses where you're mixing your heterozygote with your homozygote recessive, what you start seeing is that you're able to follow the linkage that occurs when you start looking at the predicted versus the actually observed offspring. So for instance, over here, we're taking a look at our fruit flies. And part of what we're seeing is that we're doing our test cross where we have our heterozygote mixed with our homozygous recessive. And we're interested in following the long bristles and gray body phenotypes versus the short bristles and ebony body phenotypes. And what we see happening based on our predictions and our observed number is that the chromosomes of the heterozygous parents are exhibiting crossing over. And as a result of that, what we see happening is that our recombinant offsprings are fewer in numbers than the non-recombinant offspring. And this can all be followed through by map distancing. So what you would do when you're interested in map distancing to contribute to genetic mapping is that you would go ahead and calculate your total number of recombinants, and you would divide that by your total number of overall offsprings. And that percentage would then indicate your map units. And usually what a map unit indicates is the percentage of frequency of a recombination. So for instance, if you were to come up with a percentage of 5. So if you were to do your number of recombinants over your total of offspring, and you would see that the number of recombinants are 5%, that would be 5 mapping units, which means that your frequency of recombination is 5%. And what we end up seeing when we do our genetic mapping is that as the genes keep getting further and further apart from each other, it becomes harder to follow them and their chances of crossing over, because we know that during meiosis, crossing over doesn't just happen once. You can have several crossing over events that occur within that prophase window. So our genetic mapping often tends to downplay the amount of crossing over that occurs because we can never be for sure how to account for an even or an uneven amount of crossing over. So go ahead and take a look at this PowerPoint right here. And as I was rambling on before, it basically reiterates what I was telling you, which is that as the genes are getting further apart from each other, the chances that they encounter crossing over events starts to increase. Now, when you have an even number of crossing over, that basically means that you reset the genes back to their original location. And we know that you can have multiple crossover events during prophase 1, but the thing is, when you do your genetic mapping, so when you do your Punnett square with your heterozygotes and your homozygous recessive, you can't really follow through to the exact number of crossing over that occurs. So oftentimes there are events that are not counted as recombinations, even though crossing over has occurred. And the reason for that is because the crossing over occurred in an even number, allowing the genes to reset back to its original location. So what I'm trying to say in a very long-winded explanation is that when you do your genetic mapping, usually we tend to underestimate the actual distance of the frequency and the actual map distance. In fact, what we see happening is that if the genes are further apart, the chances start getting higher and higher, and your ability to absorb it gets lower and lower. So usually what we see happening is that when you do your test cross, we basically are only about to see 50% of the recombinant offspring that are possible. So when you take a look at this illustration right here, this is figure 7.9. It shows you the mapping units on a chromosome, and then on your Y bar over there, it tells you the percentage of recombinant offspring in a test cross. What we see happening is that the line will start to flatten around the 50% mark. And that basically shows us that anything above 50% is no longer accurate to track on genetic mapping because the distance is simply too far apart and we can't account for an accurate number of crossing over that occurs during meiosis. So at the bottom right there, it says multiple crossing overs set a quantitative limit on measurable recombination frequencies. And this, of course, happens as the physical distance increases. So that is why a test cross, so when you do your heterozygous times your homozygous recessive, is expected to yield at maximum only 50% recombinant offspring. So beyond that, we can't really have an accurate prediction because the genes then simply become too far apart from each other, far apart that they can actually start adhering to the law of independent assortment. Another thing that I also want to bring up real quickly is that, and I had mentioned this very briefly in Unit 1, is that the whole event of crossing over can actually also occur during mitosis. It is obviously not as common. It is a relatively rare event. We usually see that crossing over will occur during the very early stages of embryonic development, and as the organism continues to develop, what we'll see happening is because of the mitotic recombination, so the mixing and matching of the homologous pairings of the chromosome in mitosis, the cells that have exhibit this mitotic crossing over or mitotic recombinations, these cells will have a slightly different phenotype. Now, because it tends to only occur in a handful of cells, these cells will then develop tissues that will often have what we like to call a patch phenotype. So a phenotype that is slightly different from the rest of the body. And we can, for instance, see this on this very simplistic example of our fruit fly. So all the way on top right here, we have our cells that are going to go ahead and take a look at the long bristles and the gray. Those are going to be the wild type. And the mutant form are going to be the single bristles that are coming forward, excuse me. And what we see happening is that the normal cells will go ahead and undergo their mitosis as we would expect. And very rarely, we might get some mitotic crossing over that's occurring. And if this mitotic crossing over does occur, what we see happening is it happens very early in embryonic development, And as the cells continue to do their mitotic pathway, we see that we then start generating what we call a twin spot. And the twin spot in this case is going to have the gray coloring with the normal bristles, which is different from the rest of the body. And as the organism continues to develop, we see that these cells will then contribute to patches of tissue. One will have the patch with the gray color and the single bristles, and others will have the patch with the yellow color and the normal bristles. So mitotic crossing over is not as common. It happens early on in embryonic development, and it will often cause phenotypes to appear in a patch format, which will correlate with the cells that have been exposed to the mitotic recombination. Now, let's go ahead and change topics a little bit, and let's talk about bacteria. This is the start of Chapter 9, and Chapter 9 is going to take a look at the DNA that you house within the bacteria and how are some of the different ways that we can alter that DNA. So please keep in mind that when you look at bacteria, these are usually haploid organisms, meaning they only have one copy of their genomic information, and because they are haploid, it makes it so much easier for us to track a mutation within their genome because we can do the loss of function mutation more easily, and also we don't have the dominant allele that's masking the phenotype that come into play. Now, bacteria tend to produce both asexually and sexually. When they produce asexually, that is their more common mode of reproduction. They basically do binary fission, which is that they are just replicating their DNA, and they do cytokinesis, leading to daughter cells that are identical to the original cell that started it off. However, we also know that DNA is able to reproduce sexually, and when they do that, they're able to do genetic transfer, which basically means that they swap DNA segments between individual bacteria cells. This, of course, is great for genetic variation, and we can also use it to kind of map the genes to see where exactly it's located within the chromosome of the bacterial genome. Now, what Chapter 9 is going to explore are the various different ways that we can do genetic transfer in these prokaryotic bacteria cells. And the three most popular ones are going to be conjugation, transduction, and transformation. And we are basically going to spend a few minutes taking a look at each of these. But conjugation is basically a form of sexual reproduction. It's when the bacteria cells will swap genetic information with each other directly. Transduction is when you involve a bacteriophage or a virus model to transfer DNA between bacteria cells. And last but not least is transformation. Transformation is when DNA is taken up from the outside environment, and this can be done by manipulating it in a laboratory setting, or it could be that one bacteria cell dies off, and as it's destroyed, it releases DNA that is then taken up by cells that are surrounding it closely in the environment. Here is a nice little table that summarizes our three mechanisms. So you can see the top one is the conjugation, and you can see that you have a donor cell and a recipient cell that are physically connected to each other, allowing for genomic material to be swapped. In transduction, we're going to rely on a viral model, a bacteriophage, and in transformation, we are looking at taking up extracellular DNA, and like I said, it can either come from a dead cell or we can manipulate the DNA and obviously in like a laboratory setting, put the DNA on the outside and cause the recipient cell to take it in so that it can go ahead and express new phenotypical properties. All right, so let's go ahead and let's start talking about conjugation a little bit. And what's interesting about conjugation is that there's a whole method of steps that need to be done for the bacteria to be able to transfer or share its genomic information. So the definition is right on top of the PowerPoint. It says conjugation is the transfer of DNA from one bacteria to another. It does require physical contact, as I was stating before. Not all species of bacteria are able to do this, and that is why the mode of reproduction is still asexual reproduction for many of the bacteria species. If we see that a bacteria is able to do conjugation, then usually what we see is that we call them a donor cell. And a donor cell has, in addition to its single chromosome of genetic DNA or circular DNA, it has a smaller piece of plasmid of DNA that we call the F factor. And the F stands for fertility. And the fertility factor is what we're going to allow for these cells to be designated as F positive. F positive means that it's able to be a donor cell, and the donor cell will then be able to take this F factor and basically cause it to be donated to a recipient cell. The recipient cell are often labeled as F negative because they originally lack the fertility factor and are not able to conjugate by themselves. All right, so the goal for conjugation is to take this F factor and therefore transfer some of the bacterial DNA from an F positive cell, which is going to be our donor cell, to the F negative cell, which is going to be our recipient cell. All right, so here's a look at the F factor. Keep in mind that this is an example of a plasmid. Now, a plasmid is just a circular piece of DNA. This plasmid is completely separate from the chromosomal DNA that the bacteria has. Okay, so this is just an additional piece that is in a circular format. And if you take a look at the genes that are being expressed, you're going to notice that a lot of them are indicated or starting with the T, either TRA or TRB, and the TR basically means that it stands for transfer. So it's going to contribute to the phenotypical ability for the bacteria to donate the plasmid from donor to recipient. Here is a microscopic view of conjugation. So you can see you have your two individual bacteria cells, and what they'll do is they have to make physical contact. This physical contact will be made through a little pilus. We like to call it a little sex pilus. And the sex pilus is initiated by our F positive strain, and it will allow it to make physical contact with the recipient, who is F negative, thereby allowing the genetic material to swap from one cell to the next. Now, here is a list of the molecular events that are going to need to occur in order for conjugation to be successful. And what I want to do is I just want to kind of briefly give you a nice overview of them, but I think for many of us, we're going to benefit more by taking a look at it from an illustrated aspect. So that's coming up. So just bear with me, okay? So this is what we need to do for conjugation. The first thing is that we need to have physical contact. So we need to get our F plus cell, our donor cell, to go ahead and extend the sex pilus, contact the recipient cell, which is the F negative cell, and when it makes physical contact, it's going to draw the cell closer to it. So you can notice on your notes that it says that we are going to contact and we're going to shorten. So we're going to pull the F negative cell closer to the F positive cell, up to the point that you can make a conjugation bridge, which is your actual physical contact. The next step is that in our F positive cell, we're going to see that they have a protein complex called the relaxosome. And the relaxosome is going to interact with the origin of transfer in the F factor. And it's going to allow to cut off a single strand of DNA. We're not interested in transferring a double strand. We can just use a single strand of DNA because you can always replicate it back. So what we see is that the relaxosome in the donor cell will go ahead and cut off a piece of the F factor DNA. It will separate it into a single strand, and it will go ahead and leave the protein relaxants bound, and the complex will then start being pumped out of the donor cell and being transferred into the recipient cell. Once the relaxase is back together, is into the recipient cell, what we see is that it will rejoin the piece of DNA that it brought over from the host cell. It will rejoin it to make a circle. Remember, we're all about the plasmid. And at that point, what we see happening is that the single strand of DNA will go ahead and do DNA replication so it can restore its double-stranded anatomy. And this obviously, the restoring of the double strand is also happening within our host cell. Alrighty, let's go ahead and let's take a look at this from an illustrative aspect, okay? Alrighty, so first things first, notice how we have our two bacteria cells. We have our donor cell labeled F positive because it has the F factor. So remember, F factor stands for the fertility factor. Notice how that is completely separate from the bacterial chromosome. Okay, so here's your plasmid, is the F factor. And what we see happening is that we're going to go ahead and get the donor cell to make physical contact with our recipient cell. And in order to do that, we need to go ahead and form our conjugation bridge. So this is what the sex pilot started, by reaching out, making physical contact, and then drawing it closer so it can form the conjugation bridge. The next thing that we're going to see is that within the donor cell, we're going to have the relaxosome, the protein complex, that's going to take a little snippet at the origin of transfer, and it's going to start unwinding the F factor, allowing it to go from single-stranded to double-stranded. And as this is occurring, we still have our conjugation bridge that's making physical contact. And we see that as the relaxosome gets towards the end of unwinding the double-stranded DNA, we then see that it will go ahead and make physical contact with the coupling factor, and the coupling factor will initiate the exporter, allowing for the DNA or the F factor to move from the donor cell into the recipient cell. Okay, and we can see it right over here, nicely being explained. All right, so we're not quite done yet, but we do have to switch over to the next part of the illustration. Alrighty, so we see that as the exporter continues to pump the F plasmid through the conjugation bridge, we go ahead and we see that the relaxants and the DNA complex starts entering into the recipient cell. Here you go. So now we're in the recipient cell. And in the donor cell, what's happening is that we need to go ahead and we need to replicate our DNA and our F factor. Because remember, it is a single-stranded, and we need it to go back to being double-stranded. So as you can see on the illustration right here, follow my little red arrow, we see that the F factor is replicated again, so it's back to being double-stranded. Thank you very much. And at the same time, we see that the relaxase in the recipient cell will go ahead and join the strand of DNA. Notice how it's now becoming a circular format. There's your little plasmid. And we also see that it will replicate and become double-stranded. And at the end of all this, what you've done is you've done conjugation. You've made physical contact between a host and a donor cell. And because you're able to transfer the fertility factor, the F factor, from one cell to the next, look what happens to the bacteria cell. You now have two F positive cells. So you now have two cells that are able to initiate and conclude conjugation because both of them have the fertility factor. Now, just to make sure that we're all on the same page, remember when we were doing our conjugation, we were looking at plasmids, circular pieces, most of them are circular pieces of DNA that are independent from the chromosomal DNA, okay? Independent. And it's quite common that bacteria and yeast, they will have one chromosome, but they'll have multiple plasmids that are roaming around. We also know that plasmids have the ability to carry some genes, and what we can also see is that plasmids can also be integrated into the chromosomes. If this happens, we call them episomes, and in a little bit we're going to talk about how the F factor is able to do this as well. So far, we haven't discussed that yet. We simply talked about the fact that with conjugation, you make your physical contact, and then you allow the fertility factor to move from the host cell, from the donor cell over to the recipient cell. Here's a list of the different types of plasmids that you may or may not have been or may have been familiar with or seen before. For instance, before when we were talking about our F factor, that's an example of a fertility plasmid. It allows for the conjugation to occur. We also see that we have resistance factors. These are often utilized to give resistance to things such as antibiotics and other toxins. We have decorative plasmids, which allow the bacteria to metabolize different organic solvents or unusual substances that they naturally wouldn't be able to break apart. We have the colicins, the col-plasmids, that allow for proteins to kill other bacteria, so they use it as a defense mechanism. And then we also have the virulency plasmids. These plasmids have the ability to turn a bacteria in more of a pathological strain, which obviously could be detrimental. And then you have the strain that we like to call the HFR, the high frequency of recombination strain. This strain was discovered in the 1950s, and basically what they saw is that based on the research, it said that certain fertility factors, so some of the F factors that we were just chit-chatting about, they do have the ability to be integrated into the actual bacterial chromosome. And when they integrate into the bacterial chromosome, then what we see happening is instead of just calling them F factors, we are going to go ahead and call them HFRs, or high frequency of recombination strains. So I don't want there to be any confusion. Please know the difference between F positive, fertility, and your HFR, your high frequency of recombination. So what you can do is you can go ahead and take a look at this illustration right here. We can see we have a donor cell because it's an F positive cell because it has the bacterial chromosome, and more importantly, it has the F factor, the fertility factor. Okay, so when we were just discussing our conjugation, all we were interested in is moving the F factor from one cell into the next. We were not allowing it to mingle with the bacterial chromosome. And now what I'm saying to you is that we know for a fact that you also have HFR cells, which are high frequency of recombination cells. And in these particular cells, what we see is that the F factor, here pictured in red, is able to integrate itself into the bacterial chromosome. And that, of course, is what's pictured here in blue. So whenever you see the F factor integrated into the bacterial chromosome, please don't call it an F positive cell. You're going to call it an HFR cell, a high frequency of recombination cell. And what we can see is that we can go ahead and go back and forth because occasionally the high frequency of recombination cell, the HFR cell, will go ahead and reverse the integration and cause part of the plasmid to be ejected from the bacterial chromosome. And when doing so, it can actually take some of the genes from the bacterial chromosome with it, allowing it to be transferred into another bacterial cell. So here, for instance, we have a high frequency of recombination cell, HFR, and we can see that the plasmid has been incorporated. And what's going to happen now is we're going to reverse this integration. So we're going to see that our F factor is going to loop, and it's going to go ahead and eject itself from the bacterial chromosome. And as it does so, notice how the F factor no longer looks the same. It's not all red. It's a combination of red and blue, indicating that it took a piece of the bacterial chromosome with it, along with some of the genes that were being carried. So for instance, it says right here, you have the gene for proline, which means that you're able to generate that amino acid, and also the gene for your lactose. And that was originally found in the bacterial chromosome. Notice the blue coloration. So when you see that the factor is ejected and it's now a combination of original and bacterial chromosome, what we're going to do is we're going to call it an F prime factor. Okay, so an F prime factor is an F factor that has been removed from an HFR cell, and contains pieces of the original plasmid and pieces of the bacterial chromosome. Alrighty, I know it's a lot of F's to take a look at, right? So we have the F factor, we have the high frequency of recombination cells, the HFR cells, and now what we see is that we have our F prime factor, and the F prime factor means that we have a plasmid combined with bacterial chromosomes because we had an HFR cell where we went ahead and we reversed the integration of the factor from the bacterial chromosome. So let's take a look at the slide right here. And this one says, what if you have conjugation between the HFR and the F negative strain? So you have a high frequency of recombination cell and you want it to interact with an F negative or a recipient cell. How is that going to go along? Well, we're going to use some of the very same steps that we did with our original conjugation. But what you want to keep in mind is that whenever you're ejecting a plasmid from an HFR strand, chances are that you're able to move some of the genes from the bacterial chromosome from the donor cell into the recipient cell. So let's go ahead and see what it says. It says that one of the DNA strands has to be nicked at the origin of transfer, and the origin of transfer is basically your starting point, and this will indicate the direction of the piece of DNA that will be removed and eventually be transferred. Now, any of the genes that are close to the starting point and the direction of transfer, chances are that those will move along with the plasmid as well and will be able to be relocated from the donor into the recipient cell. So we see right here on our second step, it says a strand of DNA from the HFR chromosome, so that was the chromosome that was the combination between the bacterial chromosome and the F plasmid, begins to enter the recipient cell, your F negative cell. And on average, we see that it should take about 2 hours for the entire chromosome from the donor to be moved over to the recipient cell. But oftentimes there are some interruptions which do not allow for complete transfer, so only a portion of DNA will be transferred between the two individual cells. Once the chromosomes are into the recipient cell, what we do see is that they need to be integrated into the new host or into the recipient's chromosome. And in order to do that, we have to look for a homologous region so that we can go ahead and provide possibly some new alleles to this recipient cell. I guess it's best for me to show you an example instead of me just kind of rambling off some of the steps. So let's go ahead and take a look at our next slide. Alrighty, so what we're looking at over here is we have our donor cell. Here is our HFR cell, and as you can see, we are looking at an example where our donor cell has pro-positive. That is going to be proline, so it's able to synthesize proline, and it has the lactose positive, which means it can metabolize lactose. Fantastic. Now our F negative cell, let's take a look at its alleles. It is pro-negative and lac-negative. So it's not able to synthesize proline and it's not able to metabolize lactose. Alrighty, so it could kind of benefit from some of the genes that come from the HFR cells. So let's see, how are we going to see this interaction occurring? Well, first notice that you're still going to need a conjugation bridge. So you're still going to need to make your physical contact. And then what we see happening is that we're going to go ahead and we're going to remove one piece of the DNA from the site of origin. And the origin site, remember, will indicate the direction that it's going to move. And because the lactose gene is so close to the origin site, that means that there's a high possibility that it will be carried over to our donor cell. The proline is a little bit more downstream, so that's going to take a little bit longer of the way to go. So go ahead and take a look at this picture and notice how the DNA is being, in essence, uncoiled. It's going from double-stranded to single-stranded. And as it's doing so, what we see happening is that your proline, and your lactase start to move from the donor cell into the recipient cell. And as it's moving into the recipient cell, on our previous slide, we know that it takes about 2 hours for the whole thing to move. So what we see is that we have two scenarios. We can either have the luxury of having all 2 hours, or we might have an interruption that we encounter. What's going to be the difference? Well, if we have all of the 2 hours, then chances are that we're able to move both of the proline and the lactose genes into our recipient cell, allowing for the incorporation of it into the genome, the bacterial genome, meaning that now it has two new alleles. If we have a short time for conjugation, that means that the transfer is going to be interrupted. And chances are is that the genes that were very early on in the origin of transfer, those are the ones that are really close by. They're going to be able to be transferred. But the genes that were more downstream, further down the road, those are going to get interrupted and they won't have a chance to be brought over. And in fact, if you go ahead and you take a look at your F negative cell, your recipient cell, notice how the bacterial DNA, that was incorporated once conjugation was ended after a short time, we were able to successfully bring over the lactose gene, but look what happened with the proline gene. It didn't change. And that is because it was further down the strand of donor DNA, and because conjugation was interrupted, it wasn't able to be successfully incorporated into the recipient cell. Alright, so here you go. Here's our little blue boxes. So if you have the longer segment, you can bring over more genes. So as you can see, both of them will be able to transfer. And then unfortunately, in the shorter segments, we were able to bring over one and not the other. And in case you're wondering, well, how do we pick which one we're bringing over? Well, whichever one is closer to the origin of transfer, meaning it's one of the first ones to encounter or enter into the recipient cell, they'll be the ones that will be brought over successfully. Another method that we can start changing up the genomic content in a bacterial cell is through transduction. Transduction is when we're going to use a bacteriophage as our transport mechanism. A bacteriophage is a virus that specifically goes and attacks bacterial cell. So a bacteriophage has exclusively a host cell that is a bacteria. And we've done lots of research on these before. You can look up some of the papers that they've done. Some of the most popular phages include P22, which does a lot of work with salmonella. E. coli tends to often be infected with P1. And what we're doing in transduction is that we're using the viral model to go ahead and infect the cell and enable to take a piece of its DNA so that we can go ahead and eventually transfer it over into another recipient cell or into another host cell. It all depends on what your ultimate goal of genetic transfer is going to be. Alrighty, so let's take a look at some of the steps that are involved when you do your transduction. So once again, we have our donor cell. And over here you can see we have our phage DNA. So here's our little virus. And what we're going to do, and this can happen both in nature naturally, but obviously in a laboratory setting, we can also manipulate the situation since we're familiar with the steps. We can see that the phage DNA or the virus will start interacting with the donor cell. And the phage will go ahead and do what viruses does. It will inject or infect the donor cell, and once it has access to the DNA of the host, we see that it will initiate the lytic cycle, meaning that it will go ahead and break down the host DNA in different fragments, and it will utilize it as a template so it can replicate additional phages and thereby increase its overall viral load. So you can see right here, it says the phages will start assembling. Occasionally, a phage will carry a fragment of the host cell chromosome, and in this case, with our example, we are looking at the histidine gene, which means the ability to go ahead and secrete that particular amino acid. Now, why do we care about that? Well, because what we see happening in transduction is that now we can go ahead and we can use our virus to transfer a new allele into a host bacteria that originally didn't have that particular genetic expression. So what we see happening here is that the transducing phage will then go ahead and inject its DNA into another recipient cell. And as it does so, what we see happening is that we have a chance of doing recombination. And recombination basically means that the transduced DNA, the DNA that was introduced through the viral model, is able to be recombined and interpreted or integrated into the recipient cell, allowing now for you to generate a recombinant bacteria that in this case has a new allele, the ability to synthesize histidine. Another way, or our last way of being able to move genetic material between bacteria cells is through transformation. Now, transformation by definition basically means that we have extracellular DNA that is taken up from the outside and introduced to the inside of the bacteria. This can be done either by a dead bacteria that is degrading, releasing its DNA, and being picked up by neighboring bacteria cells, but also in a laboratory setting, we can go ahead and artificially expose DNA to host cells. So for instance, in our genetic lab that we do a transformation experiment where we introduce a glow-in-the-dark gene into E. coli. And then what ends up happening is that the DNA is extracellular. We encourage the host cell of E. coli to take it up by changing the temperature and exposing it to calcium chloride. And as we do that, the DNA is incorporated into the bacterial chromosome. And when we come back a week later, what we see is that our E. coli cells are able to glow in the dark, all because they're able to have a recombinant allele and they're able to express a new phenotype due to the fact of integrating this extracellular DNA. Now, this work was initially discovered in the 1920s by Frederick Griffin, and I'm sure most of you might remember the story of Frederick Griffin when he did his work with Streptococcus pneumonia, where he had the rough strand and the smooth strand, and he basically was trying to figure out if protein or DNA was our true genetic material. And along the way, he also figured out the fact that you're able to extract extracellular DNA and integrate it within the bacteria cell. As you can see at the bottom of the slide, it does remind you that there are two types. You can have your natural transformation, which just happens out in nature, no outside help needed. And then we have the laboratory manipulation, which will then be part of artificial transformation, when we highly encourage cells to take up DNA through the use of things like temperature changes, calcium chloride. We can also do electroporation, anything that will make the membrane more permeable so that it makes it easier to take up extracellular DNA. Now, if the cell is able to take up DNA, that bacteria cell is called a competent cell. And that is because they have competency factors. And competency factors are going to be very important because it turns out that these particular proteins are going to allow you to take this extracellular foreign DNA and allow it to be incorporated into the chromosomal DNA from the original host cell. So we need these competency factors because we want the DNA from the outside to be integrated into our genome or our competent cells' DNA. And in order to do that, we need our competency factors. So here are our steps for transformation. First up is that we're going to need to have access to extracellular DNA. So as you can see, step number 1 says there's a large fragment of DNA cells that is found in the extracellular environment, and we're going to go ahead and see that they're going to start binding to the surface of the cells. And I should really emphasize that it's binding to the surface of competent cells, right? Because competent cells have specific receptors that will promote the binding of this extracellular DNA. As it starts to bind with the large fragments of DNA, we do see that it will have endonuclease enzymes that will encourage the extracellular DNA to be cut into smaller pieces. And as it's being fragmentized, we see that one strand of the DNA is degraded and the other is able to enter into the cell. There's no point in bringing the double strand in because you're able to do DNA replication. So all we really need is just a single strand of DNA and the other one will get destroyed. Now remember, in order to be successful with transformation, your competent cells is going to utilize the competency factors. So what we see happening in step number 4 is that the DNA from the outside needs to be incorporated into the chromosome of the competent cells. And in order to do that, we need to have homologous recombination. And homologous recombination basically means that we're going to look to have similar DNA strands lined up between the extracellular DNA that's incorporated and the host bacterial DNA. So we want to have somewhat of a similar DNA sequence that will line up. Now, as it lines up, it will create what we call a heteroduplex DNA. And a heteroduplex DNA is basically this awkward transition point where you're trying to incorporate the foreign piece of DNA into the genomic or bacterial chromosome strand. Not all bases will be perfectly aligned, so you might have some mismatch and some sequences that are improperly aligned, but this tends to be a temporary strand until we can utilize our DNA repair enzymes to fully integrate the extracellular DNA into the host DNA, hopefully therefore allowing for new genotypes to be expressed in our competent cells. So here we go in our illustrative example. We're going to go ahead and see that we have receptors on our competent cells that are able to detect and interact extracellular DNA. And remember, they're going to utilize those endonuclease enzymes to break it up into smaller fragments. And at one point, what we're going to see is we're going to degrade one of the double strands. So only a single strand of DNA is taken into our competent cells. All right, now once the DNA strand is inside, the next option is to make sure that it lines up to a homologous region, so a region that looks very similar on the bacterial chromosome itself. Okay, remember, we're trying to do homologous recombination, so we're looking for a piece that is very similar. So here on this illustration, we can see that the DNA from the outside and the DNA from the inside, from the genome, have lined up and they found a beautiful homologous region that will allow you to generate lysine versus the mutant that does not allow the synthesis of the amino acid. And what we see happening now is we're going to go ahead and start integrating and creating a heteroduplex. So the heteroduplex, remember, we're going to expect to have some mismatching. Notice how you have a little bit of looping because the bases are not fully interacting with each other. That is okay because we do have our DNA repair enzymes that are going to allow for a smooth integration. And if all goes well, now you have a transformed cell. You have a recombinant cell that is now a combination of the extracellular genes that were just located outside a while ago that have now been fully incorporated into the competent cell's chromosomal DNA, allowing for a new genotype to be expressed. Look at that. Beautiful. Now, when you look at these particular steps, so when we were talking about our conjugation, when we were talking about our transduction, when we were talking about transformation, all these ways of getting different sources of DNA into a recipient cell, this is all an example of horizontal gene transfer. So horizontal gene transfer is basically when you transfer genes from one organism to another. And in our example, we kind of stuck to our bacteria, but it's actually quite common that you can do different species, and you can kind of mix and match. And this is really how you create lots of hybrid or recombinant DNA species. This is also kind of like the method behind things like developing GMOs, where you have multiple sources of DNA that are sliced and spiced into a recombinant organism. If you've heard of vertical gene transfer, and you were wondering what's the difference, well, vertical gene transfer is usually when we take a look at reproduction. So it involves the transfer of genes either from mother to offspring or from parents to offspring. So if we're doing just natural reproduction and we're looking at something like a pedigree, then what we're looking at is a vertical genes transfer, so from parent to child. Horizontal genes transfer usually means that it does not have to be related. We're transferring genes from one specimen into the next. As you can imagine, in bacteria, what we see is that a lot of the genes are able to do horizontal gene transfer. In fact, one of the issues that we're having right now with the fact that some of the antibiotics are proving to be useless because bacteria is mutating and acquiring antibiotic resistance. Some of that is due to the fact of the horizontal gene transfer and the interaction of different genetic sources from the bacteria itself. In fact, if you see the signs for MRSA, it stands for methicillin-resistant Staph aureus. That is one of the main issues that we see happening when a patient becomes exposed to Staph aureus, which is a bacterial infection that you would usually get on your skin, and you would go ahead and get some penicillin or methicillin, any of the regular antibiotics, and it would get rid of it. However, because of the mutations, MRSA has developed, which means that our regular course of antibiotic treatment is not as successful as it was before. This obviously can be a very serious problem. So one thing that they're trying to do to combat it is that they're trying to regulate how much antibiotics are being dispensed. We are trying to get our immune system, in essence, to figure out a way of fighting off these antibiotic resistance bacteria. All right, let me stop talking because I find myself kind of going off topic and just rattling off about different world problems. All right, guys, I'm going to go ahead and stop here for part 1. We will go ahead and take a look at viruses in the next chapter. That'll be chapter 10. And as always, I know I'm starting to sound like an old record player. My apologies. But if you have any questions, comments, and concerns, please do not be shy. Do not hesitate. Go ahead and send me that email or join us in the discussion session, or let's Blackboard collaborate, whatever you feel comfortable with. All right, enjoy the rest of your day.